Hi everyone, we're into week 5 and we're going to start talking about antimicrobial treatments. So now that you've looked at pathogenicity of microbes, we're going to start talking about how these can be treated. And in this mini lecture, we're going to talk about the interactions between drugs that we may be given to a infected host and the microbe that might be infecting the host. So with interactions between the drug and microbe, there's a number of different goals that we have with antimicrobial drugs. It might be that we want to disrupt cell processes or structures within either bacteria, fungi, or protozoa. It could be that we want to inhibit virus replications, so stop viruses from replicating and spreading. It could also be that we want to interfere with the function of enzymes that could be required in the cell of the infecting microbe. And also we could destroy structures that are already formed in the cell that would make it no longer functional. What we're really looking for when we think about drugs is selectively toxic, so that it would kill or inhibit something in the microorganism, but not in the host cells, right? We want to be able to take care of what the microbe is, but hopefully not do damage to ourselves. So drugs that have excellent selective toxicity will be things that might block the synthesis of a bacterial cell wall. Since in eukaryotes, humans, we don't have cell walls, those would not affect us. And that is the big benefit of using something that's selectively toxic. Now, there are drugs that are toxic to humans, and those are ones that would maybe target the cell membrane, since we also have a cell membrane. And if the infectious agent is similar to the host cell, it's very difficult to achieve selective toxicity. So that's something to think about, how those prokaryotic and eukaryotic cells differ, or how human cells differ from the microbe that might be infecting it. And the goals of chemotherapy, so when we give a drug, the goal is to identify something of that living cell and either removing, disrupting, or interfering with the requirements that it has to live. And that can fall into inhibition of cell wall synthesis, inhibition of nucleic acid structure and function, inhibition of protein synthesis. Also, we might be looking at interference with the cell membrane structure and function, or inhibition of folic acid synthesis that would be needed by that cell. This is a really nice figure that shows the primary sites of action of antimicrobial drugs on bacterial cells. We have the protein synthesis inhibitors that act on ribosomes. Remember that the ribosomes in prokaryotes are different than eukaryotes, so it shouldn't damage the host cell if it's a bacterial microbe that we're after. There's also folic acid synthesis in the cytoplasm that we might want to disrupt, cell wall inhibitors, cell membrane, things that would cause a loss of that selective permeability and make it more permeable. And then lastly, DNA and RNA, we may have something that would attack those. So this is a really nice one that kind of sums it up. And then the last 2 slides are a table of common drugs and what they actually target. This may help you with your worksheet. If not all of them are here, you may need to go to an outside source to find those targets and for the worksheet that we have. Hopefully this gives you enough of a background that you have some idea about the drugs that are being used. And keep in mind that when we talk about the antibiotics or antimicrobials, the antimicrobials can also affect fungi, viruses, protozoa, even worms. So when you're looking at these, we're going to be looking at all of those. If you have any questions, let me know, or check in on the office hours and instant message me. Thanks.